Okay. Good morning, good afternoon, and good evening, everyone, and welcome to our webinar series, New Tools of the Trade in Life Sciences, Technologies that Enable Breakthroughs. Today's webinar will focus on chromatin binding of RNA polymerase II and its phosphorylation forms through the cell cycle by flow cytometry. My name is Renate Weitzbauer, and I'm the lead editor of the webinar series at Bioprotocol. And for those of you who are not that familiar with Bioprotocol, it's an open access online journal whose mission is to make research more transparent and reproducible. And it is now my distinct pleasure to introduce today's moderator for the session, Professor Antoine Desmarais. Antoine is an assistant professor at the Department of Biomedicine at Aarhus University in Denmark, where his lab investigates the mechanisms that enable adult stem cells to regenerate tissues. His work has been published in several high-profile journals, including Nature and Science, and he has received international awards for his research and leadership, including an MDA development grant. He is regularly teaching communication and resilience to science trainees and co-wrote the book Magnetic: How Great Leaders Persuade and Inspire. Antoine holds a PhD from Leiden University in the Netherlands, a postdoctoral certificate from Stanford University, and an Ignite certificate from Stanford University Graduate School of Business. He is also a Stanford certified facilitator of compassion. And with that, Antoine, thank you so much for being here today, and the floor is all yours.
Thank you, Renata, for that wonderful introduction. I would extend also welcome to everybody here to the next Bioprotocol webinar, which is a place where we profile and discuss cutting-edge technologies and advances in biomedical and life sciences. And the goal here really is to facilitate your access to technologies that enable breakthroughs in your research and improve the reproducibility of research in general. And today we're going to delve into flow cytometry technologies, as we have a wonderful speaker, Helga Landsberg, who's a senior scientist at the Institute for Cancer Research at the Oslo University Hospital. Helga obtained her PhD in 2006 from the University of Oslo in the lab of Philippe Collas. And there Helga made an important contribution to the field of reprogramming when she and colleagues reported that human fibroblasts can be reprogrammed by transient exposure to cell-free extracts. So for example, she reported that 293 T cells gain the expression of T cell receptors and transcription factor networks, gain the ability to express the interleukin-2 receptor beta in response to CD3 stimulation. Upon exposure to a neuron extract, cells gained the expression of neurofilament genes, started making polarized outgrowth. And this work really paved the way for later studies demonstrating the feasibility of reprogramming by transient exposure to RNA or protein molecules. Then she switched fields as she went on for her training as a postdoc. She started working with Randy Celiusson's lab, working on the regulation of RNA polymerase II at the interconnection between DNA transcription, replication, and repair. And she found that depletion of the CTD-associated phosphatase P nuts triggers R-loop-independent accumulation of ATR during the G2 checkpoint of the cell cycle. So more recently, including today's talk, Helga has been interested in the regulation of RNA polymerase II in the context of the cell cycle. And today she is joined by graduate student Lily Bay, who will join in for the Q&A afterwards to talk about assays to assess RNA polymerase II chromatin occupancy. With that, Helga, thank you for joining us, and we really look forward to your talk and your magnificent work.
Thank you very much. Should I share my screen? Yeah.
Absolutely.
Yes, thank you, Antoine, for that kind introduction. And today I will go through the detailed protocol for our new method to measure chromatin binding of RNA polymerase II and its phosphorylated forms through the cell cycle by flow cytometry. So through today's talk, I will give a brief background, and then I will give an overview of the protocol and go into details of the critical steps that are important for the success of this protocol. And then I will end by describing some potential uses of this protocol. So RNA polymerase II is the enzyme that transcribes DNA into mRNA. And mRNA is later translated into protein. So RNA polymerase II plays an essential role in cellular protein production. And in light of this essential role in protein production, RNA polymerase II has been extensively studied. And the RNA polymerase II transcription cycle involves the recruitment of RNA polymerase II to the promoter region, where it forms the pre-initiation complex along with transcription factors. RNA polymerase II then starts to transcribe, and upon initiation, the C-terminal domain of RNA polymerase II becomes phosphorylated on serine 5. And shortly after initiation, RNA polymerase II pauses at the promoter proximal pause site. And here, RNA polymerase II can either undergo premature termination or it can continue into productive elongation. And if it goes into productive elongation, it becomes phosphorylated on serine 2 in the C-terminal domain. So after RNA polymerase II has finished transcribing, it undergoes termination and it's released from its chromatin or DNA template. So globally, we can say that serine 5 phosphorylation is a marker for the promoter proximal pause, Pol II, and serine 2 phosphorylated RNA polymerase II is a marker for productive elongation. So why would we want to study RNA polymerase II by flow cytometry? Well, flow cytometry allows us to identify cell cycle location without the need to synchronize cells. And synchronization is important to address cell cycle effects when using methods that rely on lysates from large amounts of cells, for instance, Western blotting. However, synchronization can induce changes in transcription or replication stress. So the ability to identify cell cycle location without the need to synchronize is an important advantage of flow cytometry. Flow cytometry also gives us highly sensitive measurements by including barcoding, and I will explain this in detail later. It's also statistically powerful because it allows us to measure levels in thousands of cells per sample per experiment. In addition, it's a rapid technique, which takes about 3 days to get results. However, it measures global levels of RNA polymerase II per cell and doesn't give any sequence-specific information. So when is it irrelevant to study global effects of RNA polymerase II? Well, one example is after UV radiation, where RNA polymerase II levels are globally reduced by proteasome-mediated degradation. And this is shown here to the right, where this is a Western blot showing the reduction in RNA polymerase II levels 3 hours after UV, where the levels are strongly reduced compared to the non-irradiated cells. And this reduction is dependent on proteasome-mediated degradation because the proteasome inhibitor MG132 inhibits the reduction in Pol II levels. So this reduction in Pol II levels after UV is thought to prevent deleterious effects of lesion-stalled Pol II, for instance, due to collisions with replication and transcription, and also aberrant gene expression. However, there are other conditions where we can observe global effects on transcription. And this is an example from development and also from cancer, where you can observe hypertranscription, which is a large increase in global transcription. And this, for example, occurs during development with zygotic gene activation, where the embryo goes from a transcriptionally quiescent state, where it's dependent on mRNA from the mother, to transcribing its own mRNA and being dependent upon transcription of its own genes. And hypertranscription also occurs in cancer cells. And this is shown here to the right in this Western blot, where they blotted primary human fetal fibroblasts and compared to lysates from Ewing sarcoma cells. And as you can see, RNA Pol II phosphorylation on serine 2 and serine 5 is dramatically increased in the cancer cells compared to the normal cells. And this correlated with increased EU incorporation. So these were some examples of global changes in RNA polymerase II. But other examples are, of course, if you add an inhibitor of transcription, or you're knocking out or overexpressing a gene involved in transcription. So here I show an overview of the protocol, and it involves 3 main steps. So the first step is to prepare the extracted cells. The second step is to perform multiplex staining, and that's just another word. Multiplex is just another word for multiple parameter staining, and flow cytometry. And the output involves the quantifications and gives you highly significant differences in RNA polymerase II levels on chromatin through the cell cycle. Right, so the first step is to harvest and extract cells prior to fixation. So after harvesting the cells, they are extracted by a treatment with a mild detergent, and this leads to the release of soluble factors, while the chromatin-bound factors are retained in the cells. And then the cells that have been extracted are fixed immediately after. And I'd like to mention that detergent extraction prior to fixation by flow cytometry was first described in the early 1990s. So extraction actually removes a really large fraction of cellular proteins. And we were kind of amazed when we saw this in our blots here showing stain-free, showing total protein labeling. And here we separated bound and soluble fractions, and we kept the volumes constant so they could be directly compared. And you see that the signal for total protein is much higher in the soluble compared to the bound fraction, showing that, and when we quantified, we found that around 85% of cellular proteins are actually removed with our extraction protocol. So it follows that it's important to treat the cells gently, both before and after extraction. And we would advise you to not collect cells by scraping, because this can damage the cells. And we have observed that they are likely to clump when you perform this. So we suggest that you harvest cells rapidly by trypsin, and this is what we have done. And a trick is to, if you have short time points, for example, after DNA damage, the trypsinization has to occur at 37 degrees, but when you harvest, you can harvest in cold medium and keep the cells on ice, and this will slow down enzymatic reactions and keep your time points as accurate as possible. Right, so when we look at the extraction buffers, I just compared some extraction buffers that have been used for these kinds of protocols. And here to the left is the one that we used in our paper. And we compared it to the commonly used cytoskeletal buffer, which is often used in immunofluorescence microscopy. But the principle is the same as of extraction prior to fixation. And also the third buffer here is a low salt extraction buffer, which has previously been used for extraction prior to fixation for flow cytometry. And if you compare these different extraction buffers, they're actually very similar. And where they vary is in the presence and absence of sucrose. So both these buffers have sucrose, while this one does not. And it may be an advantage to have sucrose because it has been shown to have a stabilizing effect on proteins and protein structures. Another difference between these extraction buffers is the concentration of sodium chloride. So here in the low salt extraction buffer, you have a very low salt concentration, whereas we use a much higher salt concentration. And we find that with increasing sodium chloride concentration, this increases the stringency of extraction. So this is something that can be optimized. We would also urge you to use protease inhibitors, and also MG132, a proteasome inhibitor, if you're looking at RNA polymerase II, as we are in this protocol, because it's easily degraded and by the proteasome. And we also add phosphatase inhibitors to prevent dephosphorylation. And the extraction is also performed on ice to prevent degradation and freeze the system as much as possible. So while we were developing this protocol, we exploited a feature of mitotic cells that is that RNA polymerase II is largely lost from chromatin in mitosis, and this has been known for a while. So in an interphase cell, the nucleus is surrounded by the nuclear envelope, and inside the chromatin is in a decondensed state. But as the cell goes into mitosis, the chromosomes condense into compact chromosomes, and the nuclear envelope breaks down, and RNA polymerase II is actually released all throughout the cell. And this is observed here, and using an antibody which recognizes total RNA polymerase II. So in the interphase cells, then you can see that the DNA stain and the RNA polymerase II staining is overlapping. So it's localized in the nucleus. However, in metaphase, the RNA polymerase II staining is all throughout the cell and appears to be excluded from mitotic chromatin. And we also observed the same thing, that when in non-extracted cells, here we use a mitotic marker, which is phosphorylated histone 3 on serine 10, which marks the mitotic chromosomes. So, and the RNA polymerase stain here, you can see that it's dispersed throughout the cell and it's not overlapping with the mitotic chromosomes, also observed by the DNA stain. However, in the interphase cells, then the RNA polymerase II stain is completely co-localizing with DNA. And after extraction with the same conditions that we use in this protocol, then we can see that the mitotic cells have completely lost their RNA polymerase II staining, while the interphase cells retain their RNA polymerase II staining. So this means that after extraction, then what we are expecting in our flow cytometry charts is low mitotic staining. And this is what we observe here. So in this scatter plot, we show phosphohistone H3 staining on serine 10, which is high in mitosis. where the DNA content is high. And when we gate these cells and show them in our RNA polymerase II plot, then we see that after extraction, the mitotic cells, here shown in red, show a lower staining compared to the interface cells. And this is to the left here, we see in the non-extracted cells, then the mitotic cells show a similar staining as the interface cells, showing that RNA polymerase II is likely not being degraded in mitosis, but it's just being released from chromatin. So I told you that increasing the salt concentration increases the stringency of the extraction. And this is shown here in this figure, where we added increasing salt concentration. And this caused the mitotic population, shown here with the gated from the histone H3 positive cells. This mitotic population after extraction was going downwards, meaning that more RNA polymerase II was being released from the cells as you increase the salt concentration. So, we nevertheless decided to work with 140 millimolar salt because this is close to physiological conditions, and you can observe clearly lower RNA polymerase II staining after extraction. And to validate this assay, we wondered whether our antibody staining was similar to what was observed with endogenous RNA polymerase II. And for this experiment, we used knock-in RNA polymerase II cells expressing GFP tagged RNA polymerase II. So endogenous RNA polymerase II tagged with GFP. And indeed, after extraction, the GFP RNA polymerase II cells show a similar pattern as we observed with the antibody, with low intensity of GFP in mitotic cells, while in interface, the intensity is high. Right, so to validate the method, to make sure that the RNA polymerase II measurements were as accurate as possible, we compared the results that we got from flow cytometry with quantitative Western blotting. And again, we exploited the feature of mitotic cells with low RNA polymerase II staining, and we synchronized cells in mitosis by nicotazole treatment, and then compared the median values that we obtain by flow cytometry compared to non-synchronized cells with the values that we got from quantitative Western blotting. And by quantitative Western blotting, we mean that we have included a dilution curve, so ensuring that we obtained accurate results. So here you see that in the non-nicotazole treated cells, then most of the RNA polymerase II is in the bound fraction. And this is similar to what we see here in the flow chart. And most of the cells have high RNA polymerase II staining. However, in the nicotazole treated cells, then little RNA polymerase II is remaining on chromatin and most is released. Just by eye, it appears that the quantitative Western blotting is giving the similar results to flow cytometry. But we also verified this by performing quantifications. And indeed, we obtained similar results by flow cytometry of detergent extracted cells as by Western quantitative Western blotting. So ensuring the accuracy of our measurements. Right, so fixation conditions. So this is important, and we've we had to work to optimize it, but we found that the best thing was to fix the cells quickly by addition of a large volume of formalin at room temperature. And we tried different things. For instance, we tried lower percentages of paraformaldehyde and fixing for a longer time on ice, but this caused the loss of a large amount of cells. So really, we suggest that you try this, and this is what works best for us. Yeah, so the second step in this protocol is the multiplex or multiparameter staining and flow cytometry. So this step includes staining of cells for barcoding, staining of samples with primary and secondary antibodies, labeling ADU, and this is performed with a commercial kit, so I won't go into the details of this labeling. Staining cells with a DNA stain hex and running samples on the flow cytometer. Right, so first, I'd like to talk about barcoding, and this is something that we strongly believe in. So the barcoded cells provide an internal standard for normalization of the RNA polymerase II signal. And we find that the barcoded cells greatly facilitate the quantifications and sensitivity of the assay, and they enable all the samples to be compared to each other. And they eliminate variation due to differences in staining between the samples. So the barcoded cells are harvested in parallel to the sample cells, and they can be any type of cell. And then after chromatin extraction and fixation, they're labeled with a barcoding dye, which is generally labels proteins. In our case, we use Alexa 647. And then after the barcoded cells have been labeled, then they are added into all of the individual samples. And this is done prior to staining with primary and secondary antibodies. And this allows us to normalize. And in this slide, I show that barcoding enhances both the sensitivity and the accuracy of the assay. So here we show an experiment where we added MG132, so it's a proteasome inhibitor. So addition of MG132 will increase the amount of RNA polymerase II on chromatin, because RNA polymerase II is frequently degraded by proteasome-mediated degradation. So after MG132 treatment, then we are expecting higher levels of RNA polymerase II on staining, on chromatin. And to the left here, you see our non-treated sample cells shown in gray, compared to the barcoded cells that have been separated from our sample cells during analysis. And then they have been overlaid our sample cells in the scatter plot. And you can see that the barcoded cells are completely overlapping with the sample cells. And this is expected because they are also non-treated cells, so they are highly similar. However, in the case of MG132 treatment, then we're expecting that the treated cells should have higher staining than the barcoded cells. And indeed, it's what we are observing. With the blue cells, these are the MG132 treated cells. They are lying higher than the barcoded cells in the scatter plot. So showing that treatment with MG132 is increasing the levels of RNA polymerase II on chromatin. However, if you look at these scatter plots closely, then you can see that in this experiment, even though the barcoded cells, as I mentioned in the previous slide, are exactly the same cells here and here, they've just been added to different samples. You can see that they're behaving differently in the scatter plots. Actually, they're lying much lower here, or much lower, at least they're lying lower here compared to here. So this is differences in staining because the conditions that when we run on flow cytometry are the same, and the cells are also the same. So it has to be a difference due to staining. And so if we normalize against the barcoding cells, then this will eliminate this difference between staining of the samples. And here we show we've performed quantifications of RNA polymerase II levels on chromatin in G1 when we normalize against the barcoded cells, or using the same samples where we have not normalized against the barcoded cells. And you can see that the difference between the non-treated samples and the treated samples is increasing, and also the significance is increasing. So we strongly suggest that you use barcoding for this assay. So another important thing in this protocol is antibody staining, and the choice of antibody is critical. So RNA polymerase II, the RNA polymerase II C-terminal domain contains 52 heptapeptide repeats, 7 amino acid repeats, and they can be extensively post-translationally modified on the different amino acids, including serine 2 and serine 5 phosphorylation. And they can be different combinations of the different phosphorylations. So the different antibodies that are commercially available, or some of them are shown here. And it's interesting, I won't go through all the results here from a ELISA assay that has been published. But it was interesting, the difference in serine 5 antibodies that recognize the serine 5 phosphorylated form of RNA polymerase II. So the H14 antibody in this experiment recognized only the serine 5 and serine 2 dually phosphorylated form of RNA polymerase II, compared with the 3/8 antibody, which is the one that we use, which recognizes both the singly phosphorylated serine 5 form and also the dually phosphorylated form, including serine 5 and serine 2, or serine 5 and serine 7. So this can be an important difference because if this antibody only recognizes the dually phosphorylated RNA polymerase II, as I mentioned, serine 2 phosphorylation occurs after Pol II goes from promoter proximal pausing into productive elongation. So it means that this antibody, if this is correct, likely does not recognize promoter proximal stalled Pol II. But also the antibodies that we have used, so 3E10 and 3E8, they also have some combinations that they don't recognize as well. For instance, this combination of serine 2 and serine 5 is less recognized by this antibody. So it's just important to be aware of the limitations of the antibody. So during the validation, we validated by making sure that the antibodies were showing the expected staining pattern. So like I said, RNA polymerase II staining should be lower in mitosis. And we observed this both with the total antibody, the serine 5 phosphorylated antibody, and the serine 2 phosphorylated antibody. So this is one way to validate the antibodies. And another way to validate the antibodies is that they should be showing the expected behavior. So for example, after UV, I mentioned earlier that RNA polymerase II becomes degraded by proteasome-mediated degradation. So here, 3 hours after UV, the RNA polymerase II signal is much weaker compared to non-UV treated controls. And this is using the same antibodies that we are using. So in our assay, then we were expecting that we should observe lower RNA polymerase II levels in UV treated cells. And this was what we observed. So here, non-treated cells are overlapping with the barcoding cells, like I previously showed you. However, the UV treated cells, here shown in yellow, are lying much lower than the barcoding cells. So we were confirming that this method was showing the expected behavior of total RNA polymerase II. So another way to validate the antibody was by using transcriptional inhibitors. So here showing DRB. So DRB is an inhibitor that inhibits the transition from promoter proximal stalling into productive elongation. And like I mentioned earlier, promoter proximal Pol II should have high serine 5 phosphorylation, and productively elongating Pol II should have high serine 2 phosphorylation. So if we add the DRB inhibitor, then we should be expecting lower serine 2 phosphorylation, while the serine 5 phosphorylation should remain high. And indeed, this is what we are observing with our antibodies, that the phosphorylation of serine 5 remains high after DRB treatment, while the serine 2 phosphorylation is strongly decreasing compared to the barcoding controls. Right, so another inhibitor of RNA polymerase II is THZ1, which inhibits an earlier stage of the transcription cycle, and it inhibits RNA polymerase II prior to phosphorylation of serine 5 and serine 2. So after THZ1 treatment, we are expecting lower phosphorylation of serine 5 and lower phosphorylation of serine 2. And again, this is what we are observing in our Pol II scatter plots, that we are seeing lower serine 5 phosphorylation compared to the barcoding controls, and also lower serine 2 phosphorylation compared to the barcoding controls. Right, so cell cycle, why does it matter? Well, RNA polymerase II is influenced by the cell cycle, and the prime example of this is, of course, the much lower staining of RNA polymerase II in mitotic cells. It's not completely absent, but it's much, much lower than in the interphase cells. And another example where RNA polymerase II is influenced by the cell cycle is in when it undergoes conflicts or collisions with replication. So transcription replication conflicts occur as transcription and replication occur on the same substrate. Excuse me. And this can influence your results with RNA polymerase II through the cell cycle. And another way that the cell cycle can influence your results with RNA polymerase II is that the loading increases through the cell cycle. And this is shown here. So here we have isolated individual cell cycle phases based on EDU incorporation and DNA content. And we find, and EDU is a nucleotide analog that is incorporated into the cells as the cells replicate their DNA. So G1 cells, which are non-replicating, they should have no EDU uptake, whereas the S-phase cells, the early S-phase cells should have be positive for EDU, but have close to G1 DNA content. Cells in mid S phase should have a DNA content which is between the G1 and the G2 cells and be positive for EDU. And the cells in late S phase should have be positive for EDU and have a close to G2 DNA content. And finally, the G2 cells should be negative for EDU and have a G2 DNA content. And these cells here below, these are probably the mitotic cells because we have found that they have low RNA polymerase II staining. So after gating the cell cycle phases this way and measuring the Pol II levels with the different antibodies in our assay, we find that the RNA polymerase II levels are increasing, steadily increasing through the cell cycle. And this is expected because as the cell grows, then it becomes bigger and it has more signal. And it also DNA is replicated. So there's more DNA to transcribe for RNA polymerase II. So it was completely expected that the levels of RNA polymerase II should be increasing like this through the cell cycle. But this effect could alter your results if you're looking at the total RNA polymerase II population without taking cell cycle into consideration. Then higher Pol II levels could be caused by accumulation of cells in specific cell cycle phases. Cell cycle effects can influence your results. And this is shown here in this from an experiment where we wanted to see whether total Pol II levels were increasing on chromatin because we observed higher phosphorylation of serine 5 at early time points after UV. And we wondered whether it was caused by increased initiation or if it was caused by a signaling effect, just causing hyperphosphorylation of Pol II. And if it was increased initiation, then we should be able to observe increased levels also of the total form of RNA polymerase II on chromatin. And as you can see, we were able to see increased levels of RNA polymerase II on chromatin after UV in the G1 cells, but not in the S phase cells. So this suggests that replication might be promoting more removal of RNA polymerase II on chromatin, but nevertheless, then the increased phosphorylation of serine 5 is probably caused by increased initiation. Also, including cell cycle analysis can reveal novel findings. So this is exemplified here, where we looked at different cell cycle phases at 2 hours after UV. And we could observe that serine 5 phosphorylated Pol II was mostly removed, or was more, the levels were lower on chromatin in S phase compared to G1 or G2 phase of the cell cycle. And this was, in fact, when we added MG132 to the assay, we saw that this was caused by increased degradation of serine 5 phosphorylated form of RNA polymerase II after UV in S phase compared to G1 and G2, suggesting that transcription replication conflicts are increasing the removal of RNA Pol II from chromatin by degradation. On the other hand, serine 2 phosphorylated RNA polymerase II was more stable in S phase compared to G1 or G2 phases after UV. So this is an interesting effect that we don't really understand, but it could indicate that DNA repair is different in S phase compared to G1 and G2 phases. Right, so this brings us to acquisition, so flow cytometry acquisition. And we used an LSR2 flow cytometry from BD with the parameters Alexafluor 488 for the RNA polymerase II, phosphoserine 5 and phosphoserine 2 forms. Alexaflor 647 for barcoding, Alexaflor for 594 for EDU, and then hex for DNA, and then side scatter and forward scatter for granularity and cell size. So the first step during analysis was to separate the sample from the barcoding cells. And first we want to identify the single cells. And the single cells or the duplets can be separated from the single cells by the width, which will be bigger for when several cells are clumped together. So this allowed us to identify the single cells. And after this, we gated the sample cells from the barcoded control cells. And this was done by looking at the Alexaflor 647 staining compared to the 594 staining. So the barcoding cells were clearly higher for 647, and this could be easily separated from the sample cells. So I told you about one way to identify the cell cycle phases, but there are you can also identify the different cell cycle phases based on just the DNA content alone. So in this case, the G1 population can be identified by taking the summit of the G1 peak and then getting all the cells to the left of the G1 peak. And this will limit the amount of S-phase cells in your G1 population. And the G2 cells can be identified by identifying the summit of the G2 peak and then gating all the cells to the right of the G2 peak summit. And again, this will limit the amount of S-phase cells in your G2 population. And the S-phase cells will have a DNA content between the G1 and the G2 cells. Right, so after having identified the different cell cycle phases, then you can analyze the RNA polymerase II levels in the individual cell cycle phases. So here you see the different cell cycle population and how they locate in the RNA polymerase II plot. Right, so in the flow cytometry experiments, then bleed through or overlap can be an issue. And we wanted to avoid compensation. So we wanted to ensure that we did not get any bleed through of the 647 or 594 stain into the 488 measurements. And we verified this. You can see it here. So the barcoding cells should have high 647 staining, but nevertheless, they are overlapping with the sample cells in the RNA polymerase II plot, showing that the 647 staining in this example is not influencing the 488 staining. And similarly, the sample cells can be separated from the barcoded control cells. And here you see we've added EDU to the sample cells, while the barcoded control cells are EDU negative. But nevertheless, in the RNA polymerase II plot, then the sample cells and the barcoded control cells completely overlap, showing that the EDU staining is not affecting the 488 staining. So we strongly believe that we have very little bleed through or overlap of the different 647 and 594 into our 488 staining. But if you change the parameters or change the fluorochromes this may become an issue. So this is something to be very aware of. Right, so this leads us to the third step, which is the output. And here I show the quantifications that, or the calculations that we did, which were quite simple. So we use G1 levels as an example. So here we measured the median levels in our G1 sample, and we subtracted the median level of the secondary antibody control, which we always include in our experiments. And then we normalize against the median level in the barcoding cells. And the second equation gives us the changes in RNA polymerase II levels in G1 after treatment. And this here we get from equation 1, the RNA Pol II level in the G1 in the treated sample. And we divide this by the RNA Pol II level in G1 in the non-treated sample. And we always include a non-treated sample as one of our samples to be able to compare against our treatments. Right, so this leads us to the potential uses of this protocol. And we think it can be used to study the transcription cycle. It can be used to screen or test how RNA polymerase II transcription is affected by a specific stimuli or inhibitors. It can also be used to study different levels of transcription, for example, hypertranscription, or it can be used to study effects of individual genes involved in transcription. However, we view it as a complementary technique to techniques such as sequencing-based techniques, because it does not provide any sequencing sequence-specific information. Right, so I'll show you an example of how we used this technique to study the transcription cycle. And in this case, we added, again, we added the proteasome inhibitor MG132 in HeLa cells, and we measured the fold change in the presence and absence of MG132 and quantified this in the absence and presence of UV. And what was striking for us, especially in the absence of UV, is that it was the serine 5 phosphorylated form of RNA polymerase II, which was most enhanced by MG132, while the serine 2 phosphorylated form, for instance, after in the absence of UV, was very little degraded, or it was very little increased by MG132 treatment. And this was actually quite surprising, because at the time, promoter proximal Pol II was previously thought to be protected from proteasome-mediated degradation. And furthermore, we found that after UV, then it was not only the serine 2 phosphorylated form, which is the form that travels the most along the DNA strand, and it's most likely to encounter the bulky DNA damage directly. It's not only the serine 2 phosphorylated form, which was being increased or was being degraded by proteasome-mediated degradation, but also the serine 5 phosphorylated form was being more degraded after UV. So then we hypothesized that perhaps actually promoter proximal Pol II is degraded. And to address this, we added the inhibitor DRB. Again, this inhibitor inhibits the transition from promoter proximal pausing into productive elongation. So it has been shown that this, by ChIP-seq experiments, for instance, that this leads to the accumulation of serine 5 phosphorylated RNA polymerase II on the promoter proximal pause site. So if the promoter proximal form of RNA polymerase II was being protected from degradation, then this DRB treatment should not induce any degradation. However, we found that when we treated samples with a combination of DRB and MG132, the proteasome inhibitor, we found that RNA polymerase II after DRB was being reduced, so lower levels of total Pol II on chromatin after DRB treatment. And this was completely rescued by the MG132 treatment, showing that the reduction in Pol II levels after DRB was caused by proteasome-mediated degradation. And this was caused by a degradation of the serine 5 phosphorylated RNA polymerase II, because the levels of serine 5 phosphorylated Pol II were being strongly enhanced by treatment with DRB and MG132 compared to DRB alone. So these results showed that promoter proximal Pol II actually is being degraded on chromatin, and this is in the absence of UV, but it has also been shown that degradation by others, that degradation of promoter proximal Pol II can occur after UV. Right, so if we compare this technique versus sequencing-based techniques, we find that this technique is both rapid and inexpensive. It's sensitive and quantitative. It allows single cell analysis and cell cycle determination without synchronization. However, it lacks sequence-specific information. And if we compare this technique to, for instance, Western blotting, we find that it's more sensitive and statistically powerful. Again, it gives single cell information versus a lysate from multiple cell cycle determination. However, it lacks information based on molecular weight. Right, so I'd like to also show some results that we got with a transcription inhibitor called Triptolid. And this inhibitor acts at a similar point in the transcription cycle as THZ1. So it inhibits both serine 5 and serine 2 phosphorylation. And the reason that I show these data is because I once got at a question at a conference whether the differences, the large differences in RNA polymerase II loading in the mitotic versus the interface cells could be caused by the large chromosome or chromatin structural changes in mitosis versus interface. However, here we show using a triptolid that both total RNA polymerase II, serine 5 and serine 2 phosphorylated forms of RNA polymerase II have strongly decreased staining after triptolid treatment. Both in the especially in the interface cells, of course, because they're the ones that have the Pol II staining. So this means that very low Pol II staining can also be detected in the interface cells. So the low Pol II staining in mitosis is not a consequence of altered chromatin structure in mitosis versus interface. And also we have also verified that triptolid does not push the cells into mitosis. And here are the quantifications of RNA Pol II levels on chromatin and S-phase confirm that triptolid causes these very low levels of all the different forms of Pol II on chromatin in interface. Right, so I see that it went a bit faster than I was expecting, but I'd like to end by acknowledging the other people involved in this work. I would especially like to acknowledge PhD student Lily Bai, who has done an excellent job developing and optimizing this protocol. We work in the group of Randi Siljuasen, who has lots of experience with flow cytometry. So she has developed assays, screens, and published many papers using flow cytometry. We're also grateful to the other members of the Siljuasen lab for having a really great working environment. Trond Stokke, he was the head of the flow cytometry core facility here at the Radium Hospital for many years. He very recently retired, but he was also the first author on the paper who first described this pre-extraction prior to fixation for the use in flow cytometry. So we are very honored to have him as a co-author on our protocol paper. And we're grateful to him for this. And we're also grateful to Jørgen Martijn and Barbara Stur from the Netherlands for providing us with the knock-in RNA polymerase II GFP cells. And also to the other members of the flow cytometry core facility, which will continue being good as Eden has taken over the charge now. So thank you.
Thank you, Helga, for a wonderful presentation. As mentioned in the chat, you're more than welcome to post any questions there or in the Q&A. We do have some questions coming in online through the website, but I would start with one of my own. I was really intrigued by the observation of the signal difference in the barcoded cells from one run to the other. Could you comment on why that might happen? And is that something you would recommend that we always do for flow cytometry experiments?
Yeah, so the answer to that is that if you're measuring why it occurs is probably because of staining differences from one sample to another. So we try to be as precise and accurate as possible when we add volumes and antibodies and so on, and use the same antibody dilution for all the samples. However, staining differences can occur. It can also be caused by a very, very high staining in your samples, which might make less antibodies available for staining of the barcoded cells. These kinds of effects can be present. So, those are the reasons why there might be differences in staining from one sample to the other. And it's my opinion that yes, if you're looking at small differences and looking at median levels in accurately measuring median levels in flow cytometry, it's a good idea to always include barcoding. But not if it's an on-off situation. For instance, if you're looking at a population that's clearly much, much higher than your main population, then you might not need barcoding. It's really if you're measuring small differences in a very, very accurate manner.
So subtle differences. Well, thank you for highlighting that. To pull some questions together, you work a lot with antibodies, right? And of course, then your assays are as good as the antibodies you got. Two questions related to that. One is you put a lot of effort in antibody validation. Is that something you recommend people do for their own experiments?
Yes. So I would definitely recommend that any time if you use a new antibody, it would be important to validate it. So if it's, for instance, you could knock down or out the protein and see if the staining goes away. It's one way to validate. But yeah, we recommend validating the antibody for sure.
Don't just trust what the vendor claims, right?
Definitely.
A related question to that is what you've been doing is working with cultured cells. I imagine a lot of people will want to purify cells, maybe from primary tissues. So then it adds the cell surface marker staining panels. Have you worked with that? Could you comment on how that might work?
Yeah, so that leads into kind of increasing the parameters in your assay. I guess that's what it means. I think you would have to verify that your panel is still valid after extraction, so that your markers are still present after extraction. And another thing is that if you want really accurate measurements of Pol II, and you're increasing the amount of parameters, then you'd likely have to use compensation, which you can use, but it may increase the spreading of your signal. So if you have small differences, they might be hard to observe.
Yeah, thank you for commenting on that. Obviously, if you were to do a purification and you lose a lot of negative gates, ultimately you would ideally have only one marker additional show up, right? The positive marker for which you sort your cells. So it seems like it's a feasible thing. But...
Yeah, but your markers would have to be present after extraction. So if there's cell surface markers and you're using detergent treatment prior to extraction,
you might lose them.
You might lose them. Yeah.
Yeah, very good point. You mentioned that. Yes.
Sorry. No, I was just thinking that if you have a marker on the surface of the cell that you want to sort your sample based on that, maybe prior to extraction, then you can sort your cells based on that marker and then perform the chromatin extraction afterwards.
That's a good point.
Good idea. The protocol you mentioned using protease inhibitors, proteasome inhibitors. What about RNase inhibitors? Would RNAs impact polymerase binding?
Yeah, it's a good idea. It's a good point. So we have added RNase during extraction, and we do find that a lot of factors are released from the cells, but maybe not so much RNA polymerase II though, which would we would imagine is bound to chromatin, so mostly bound to the DNA.
Yeah,
it's a good point. For many other factors, it would be important.
Because at the moment we're looking at RNA polymerase II. Could you imagine using this method for Pol I or Pol III?
Yeah, definitely. So I haven't worked with those polymerases myself, but yeah, that would definitely be feasible. And actually, any transcription factor, this method could be used, and it has also been used before for transcription factors.
Again, it depends on the availability of your antibodies, right? And you can deploy it. I'll tag in another question of my own. All the cell cycle analysis, I noticed one cell cycle state was missing, and that is the G0 or quiescent state. Have you done any experiments looking at cells that have temporarily withdrawn from the cell cycle?
No, we haven't. So we use HeLa cells, and they're pretty much proliferating all the time, right? So we haven't. We've used some other cell types, but no, we haven't specifically looked at G0 cells, but that might be interesting.
Yeah, then it wouldn't immediately work, but that's a curiosity of mine. So thank you for highlighting that. Maybe a follow-up question is could you use this assay in some way on nuclear extracts?
I guess you could use, say you're thinking about, for instance, after reprogramming, or I guess you can perhaps extract your cells and then treat them with some kind of extract prior to fixation. It's possible. Yeah.
I think it's more about if you have tissue in the freezer, and that you extract all the nuclei from that to get a measure of.
Yeah, so probably
obviously
not.
you would lose mitosis as a readout, but
you would lose mitosis, and the other thing is that I think that would probably lyse your cells. So you need your cells to be intact, and just like I said, just scraping the cells will harm them enough to make this assay really difficult. So probably not from lysates from frozen down cells if they haven't been frozen in, for instance, DMSO to conserve the cells.
Right. Another question coming in is how easy is it to adapt this assay, or is it at all possible to say things about elongation speed?
Yeah, probably I wouldn't use it to say anything about elongation speed because it doesn't look at sequence specific information. So that would kind of be an extrapolation, maybe too big extrapolation. Yeah.
You could sort of approach it maybe a little bit with a different phosphorylation size, right? But
yeah, I guess you could say something about the maybe the stability, but still you don't know where the serine 2 is located. So I think it would be really difficult. I would be interested to see if someone is able to do that.
All right, let me check here. So maybe one last, I mean, you went over this, but could you again list the advantages of using flow cytometry to go at this, and maybe in that touch upon whether this assay could be used for any signaling event, or is it strictly something to do with chromatin associated proteins?
The advantages of flow cytometry. So I guess the major advantage would be single cell analysis, the ability to look at cell cycle location without synchronization, quantitative strength, sensitivity when including barcoding. Yeah, I don't know if I'm forgetting something now, but another thing, yeah, so any signaling event. So that's a good point. So we already showed during this work, I already showed that you could use it to look at histone H3 phosphorylation. And I guess you could use it for most chromatin binding proteins. Especially if their activation occurs when they bind to chromatin, or they're released from chromatin. In situations like that, then it's really useful. However, for soluble fraction or soluble factors, then you're losing them during the extraction. So probably not so useful for that.
All right. Thank you for answering all of these questions. If there's any more questions, do we have a way for viewers to post those?
Yes, we do. All right. Yes, so if we have more questions, you can visit our webinar website, where we have a Q&A section where you can still post questions after the webinar. And you can also participate in the discussion in this section. All right. Thank you so much for an interesting talk, Helga, and the lively discussion. I think we had sufficient time to address all the questions, and I hope this has been very helpful for everyone. Thank you again. And as I just mentioned, if you have more questions, you can go to the webinar website where you can utilize this Q&A section to post more questions. As I mentioned earlier, this website is being recorded and will be posted on our website later. This can take about 1 to 2 weeks until it has been uploaded to the website. And lastly, I would like to announce our next webinar. Thank you. Isolation and Characterization of Endogenous Nucleopore Complexes by Javier Fernandez Martinez, narrated by Samson Obado, on February 15th of next year. If you want to know more about this webinar, please visit our website. And thank you again for joining us today, and hope to see you next time.